In our lab, we are both amazed and intrigued by how evolution shaped the history of life. And so we want to understand one of the most important evolutionary transitions in life history, which is the origin of multicellular animals from their unicellular ancestors. Specifically, we want to understand the molecular mechanisms involved on the unicellular to multicellular transition that gave rise to animals or metazoans. And how do we address this question? By using a powerful scientific methodology, which is comparative genomics. So what we do is we sequence the genomes of the closest unicellular relative to animals, and we compare those genomes with the genomes of animals. By comparing the genomes of the unicellular relative with the genomes of animals, we can infer how the last common unicellular ancestor looked like. We can infer the gene composition and the genomic architecture of the ancestor. This is providing important insights. So, for example, we have found that the unicellular ancestor of animals already had a complex repertoire for genes involved in multicellularity, genes involved in cell addition, cell communication, cell differentiation. So our next goal is to try to understand what are those genes doing in the unicellular ancestor and how were they co-opted for the new functions. I strongly believe that this approach will provide important clues not only on the origin of animals, but also on how evolution works.